Hello, Vinita. Can you hear my voice?
Sir, good morning, sir. Yeah, we can
Good
hear
morning.
your voice,
Here.
sir. We
Good
can
morning.
hear.
Good morning. Can you hear my voice
Yes,
clearly?
sir. We can hear. We can hear.
Okay. Is it okay?
Yes, yes. It is okay, sir.
Okay. Nice. Then okay.
Okay. Sir. We'll just wait for 5 minutes for the participant to join, sir. Then we'll begin. Ma'am, shall we start, ma'am?
I think only 28 participants are there, no? Nita
Yes.
madam.
Shall we wait,
Yeah,
no?
yeah, we can start. It's already 11:37.
11:30. Shall we start?
Yeah, we can start.
Sir, you can unmute, sir.
Yes.
Uh, Good morning, everyone. Once again, I welcome you to the e-FDP program organized by Department of Pharmaceutical Quality Assurance, Sarojini Naidu Vanitha Pharmacy Mahavidyalaya. I'm glad to introduce Dr. Linga Banot, who is a senior scientist in CSIR IICT Tarnaka, Hyderabad. Sir, Dr. Linga Banot is having a distinguished academic background with BPharm graduation from University College of Pharmaceutical Sciences, and he has cleared it in 2006, MPharm from Naipur Mohali in 2008, and PhD from Naipur Mohali in Pharmaceutical Biotechnology in the year of 2013. Sir has joined in various industries like Vanberg Limited from 2013 to 2015, and he has also worked in Cipla from 2015 to 2019. From Jan 2019, sir joined in CSIR IICT as a scientist, and he has awarded with position of senior scientist in January 2023. With his 12 years of research experience, Dr. Linga has made significant contribution to the field of pharmaceutical biotechnology, biocatalysis, organic synthesis, etc. Dr. Linga has authored nearly about 40 research publications in high impact Scopus journals, filed several patents, and actively participated in various national and international conferences. And sir, has also guided several research scholars towards successful completion of their doctoral and postdoctoral studies. Last but not the least, sir has honored with some of the famous awards like Junior Research Fellowship Award from DBT in the year of 2008, Senior Research Fellowship Award from DBT, Department of Biotechnology, India in the year of 2010, Young Scientist International Travel Grant Awards from DBT as well as CSIR India in the year of 2013, CSIR Nehru Postdoc Fellowship Award in January 2014, Above and Beyond Award from CIPLA, Bangalore in 2017. API R&D Innovation Award in February 2018 from CIPLA Private Limited. And the most prestigious Dr. APJ Abdul Kalam Excellence Award in the year of 2022. So, with this brief introduction, now I would like Dr. Linga Banot, Senior Scientist, CSIR, IICT, Tarnaka, Hyderabad, to deliver his lecture on the topic towards sustainability and design and synthesis of efficient cascades of cascade processes with enzymes. You're welcome, sir.
Thank you, Vinita, for your kind words and thank you for your principal and all the organizers giving me opportunity to deliver lecture. And can you share my slides? Or
Will you share or should I share, sir?
Shall I share myself?
Yes,
Yeah,
sir. You can share, sir. You can share your
share.
slides.
So can you see slides?
No, sir, it is not visible.
No, sir.
Not visible.
Yeah, now it is visible, sir.
Now
Better
it
to
is-
put in
now it is
presentation
visible?
mode. Slideshow.
Okay. It's
Put it in
clear?
slideshow, sir.
So moving slides now.
You put it in slide share modes.
The slide share mode only I put. But moving or not moving slides?
No.
Not moving?
No.
Sir, no, sir. In bottom, where it is like where it is 100% it is written. No, there you just click on, sir.
Now? No moving?
Slideshow, presentation mode.
Presentation mode lo better.
Yeah, I kept in presentation mode only.
But the slides are not moving.
Otherwise, from your side you can share. Otherwise, no problem.
Okay then.
You can. Just now I send presentation now.
Yeah. Is it visible, ma'am?
Yeah, it's visible.
Yes.
Okay. Okay, sir. Now you can continue.
Thank you for giving me this opportunity, and thank you, Vinita, your kind words. And today I am going to discuss about towards sustainability, designing and efficient cascade process with enzymes, how the enzymes are more effective nowadays. So we will see in my slides. Actually, go to the next slide. Before going to the enzymes evolution, all these things, I would like to introduce the chirality and its biological activity. Most of the APIs exist as a chiral molecules as either enantiomer. So this enantiomer, one enantiomer have desired effect. It's called eutomer. Another have sometime it may be less active. Sometimes it may be different pharmacological action. Sometimes it's a totally opposite pharmacological action. So or sometimes it may cause some adverse reaction, toxic reaction. So that's why the single enantiomer is a very important. And here we are giving few examples. Just click on the slides. So here one propanol is a beta blocker drug. So it's propanol is a more potent, but S propanol is a less active. But less active also we need the single enantiomer propanol. Why? Because Yes, propranolol, it metabolite may leads to metabolite leads to causes effects on our organs. So that's why single enantiomer is very important. The next example here, just click, and then here one intermediate, the quinine and quinidine, but one quinine shows the anti-malarial, but in the opposite enantiomer shows the anti-arrhythmic. Here total different pharmacological action. Next slide. And if you see the dobutamine is a alpha antagonist action. Here R dobutamine, yes, dobutamine alpha agonist action. Totally different pharmacological action. The enantiomer is both the enantiomer, but it's a total different opposite action, opposite pharmacological action. And when see some list in table here given ethambutol, ketamine, dopa, penicillamine, asparagine. Ethambutol we use for tuberculosis, but it leads to blindness, and ketamine when we use for anesthesia, it's used to hallucinogen, and like that. If you see the asparagine, the S forms a bitter taste and R form is a sweet taste. So that much difference is there between this enantiomer. So enantiomer and coming to the thalidomide. The thalidomide is when tragedy happened when thalidomide used for desired sedative effect in pregnancy women's, actually that time most of the baby born without limbs and teratogenic effect it's happened is because of yes thalidomide. So that's why US FDA, go to the next slide. That's why US FDA as well as European agency both based on that thalidomide tragedy. If it is a drug is enantiomer, it should be study on that enantiomer, which enantiomer is the more active, and what about the another enantiomer, either it is inactive or it will be cause some a toxic effect, or it may be cause some other pharmacological action. So it should be needed to study and take approval only desired one like etomer. So in that way, from the 1992, US FDA approved the single enantiomer, as well as European Union also 1993, but it affects from 1994. Both based on this implementation, we can see the next slide. We can see next slide. We can see the just this is the this slide, just US FDA approval drug. There three categories there. One is Achiral in red color, and then single enantiomer in blue color, and Resmic in white color. And you can see here, actually, it is approved from US FDA from year wise, the graphical representation. And the blue line, most of the drugs in the blue line only. So, and then when we see the chiral Resmic one, very less. Actually, in the 2010, the Resmic is 0, and 2012 is 0, then to 14, and then 16, 17, then 19 also it's 0. But it's approval very less, 1 or 2 molecules. So this much importance is there. Then next slide is a go for next slide. Or can I change slide from my here? No problem. Next slide. Hello. In the next slide, actually, European Union approval drugs.
It's not changing here.
So it's not changing. Next slide.
It's not going to next slide.
Not going.
No.
You are in slide 5, sir.
Yes.
Go for slide 6.
Sirisha ma'am, once check it. No, it's not changing.
No, sir, it is not changing.
What
Actually, you have from your end only you have to do, sir. You are only sharing.
No, you people share, na? That's why from my.
Yes, sir. You have only shared, sir.
It's changed now.
Okay.
Changed, sir.
So this is okay. Keep in number 6 slide. Yeah.
Yeah.
So here European Union approved NASS approved drug from the 10-year duration. So here also in yellow chiral, and then blue in single enantiomer, and white in the Resmic. If you see the from 2017 onwards, there is a single none of the Resmic drugs are approved here. Only single enantiomer is approved. So go for the next slide. So based on importance of biological activity of that chiral molecule as well as approvals of drugs from US FDA and European agency is a huge required the synthesis of the single enantiomer is very important. For that, different methods are there for chiral pool synthesis or racemate resolution, either kinetic or crystallization method or prochiral substrate or symmetric synthesis. These are the different methods are there, but today we each method have their own advantages as well as disadvantages. But we will discuss in our presentation in enzymatic kinetic resolution, in resolution and other prochorals with biocatalyst. So we will go ahead with those things. And next slide. So before going to that, so what are the reports are there in chemical synthesis of this aralaxypropylamine? This aralaxypropylamine is also a very important molecule for the most of the beta blockers, as well as some integral part of the bioactive compounds and other than integral part, anti-cancer, antiviral, muscle relaxant, cup suppressant. So many intermediates are there for that aralaxypropylamine enantiomer. For that synthesis of different chiral synthons, they can use from the literature, the chiral synthons are using to synthesis that aralaxypropylamine, that they can use D-mannitol or sorbitol. But here, chiral synthons, there is a required, first criteria is requirement of the resemblance with the naturally available substance required. And getting the resemblance is a very difficult. If you get also, what will happen? When we get this resemblance also, it's take so many steps, many synthetic steps are necessary to produce a single enantiomer of aralaxypropylamine. So when we increase the number of steps, it's we lose the yields. And coming to the chemical chiral catalyst, their chemical chiral catalyst also they can use a titanium alkoxide or rhodium Jacobson catalyst, the ruthenium Jacobson catalyst, meaning the ruthenium argonum, or these are the metal catalysts. These are the metal catalyst with chiral ligand. A metal catalyst, when we do the chiral ligand, these are the expensive. And once we perform the reaction, we can't regenerate these things. And then regeneration, as well as when we perform the reaction, after completion of the reaction, we have to prove that each metal, whatever titanium, rhodium or ruthenium, each metal, it will be in ppm level. So for the ppm level, when we purify the final molecules, so we may lose yield, and however, we lose yield also. Again, what happened now? The chiral selectivity is also most of the times very less. So selectivity is less, expensive, and purification is difficult. So these are the and further it's expensive. So a lot is a problem with chiral chemical catalyst. So go for the next slide. So, as in the conventional chemical process, what will happen now? Actually, from these are the hazardous chemicals. Sometimes we perform the reaction at pressure conditions, and we also read the news from some of the times there's some pressure reactor is blast like that. So actually, if we want to do hydrogenation, or definitely we have to do with palladium and some flammable reagent in pressure condition using hydrogen gas. So, and generation of the waste also too much in conventional process. So just click on the slide. So when we the nowadays in the recent days, actually enzymes are evolved very very well. We go through the next slides so we can see how the evolution happened in enzymes, and the waste generation also very minimal as compared to the conventional method. So here biocatalysts took very prominent for the synthesis of single enantiomer. Next slide. So what is a biocatalyst? The biocatalyst is like that. So it's a biological system to catalyze the conversion of one compound to another. And here we can use a wholesale or cellular extract or isolated purified enzyme can mobilize to get convert from one compound to another compound. And in the biocatalysis, actually recently the one paper in Nature, it's a Nature review came that you categorized into three waves, the first wave and second wave and third wave. Based on that, actually in the first wave, we use wholesale as a biocatalyst for biotransformation of the most of the steroidal biotransformation or penicillin production, and then from the penicillin 6-APA to synthesis of semi-synthetic most of the antimicrobial as well as some antimicrobial products also from biotransformation we produce, and that's the first century ago to 1980. But in the 1980 to 1980 second wave, they given the second wave. Why? How? By because there some molecular biology, protein engineering, advanced technology some came, and then that's why they characterize the gene sequence and which is the gene for producing that specific enzymes. They cut the gene enzymes and then clone to some vector, and once the clone, that vector will transfer to an E. coli, and production of that particular enzyme is more, so perform the reaction and very well. And later in the third wave, coming to third wave, from the third wave, the 90s onwards, what will happen actually in bioinformatic tools also, so many advanced tools came, novel tools came from in bioinformatics, and as well as molecular biotechnology, molecular biology also, so many advanced DNA technologies tools came. So both this combination of molecular biology as well as bioinformatics, both the tools they can use, and they develop the enzymes such a way. Actually, if it is a molecules, it will not bind to that specific enzymes, so they can change that catalytic of that, so binding nature. First the bind that one, then it is conversion rate is if it is a conversion is around 60-70%, they can improve the conversion. So in that such a way they modified the twist, they change the amino acid, and then which are the amino acids to inhibit that conversion, so then modify, and which amino acid they can modify it from the bioinformatics they get it, and then modify it, then improve that strain, the conversion. And not only conversion, for industrially we need some, most of the compounds are insoluble in buffer and water, so we need some co-solvent like DMS or like that solubility purpose. So solvent resistance also they improved. Not only solvent resistance, and the we need to do some gram scales per liter per 100 gram, like that. So for that substrate inhibition is take place, that substrate inhibition resistance also they overcome. And then up to if you want to do some temperature and pressure, all these things, temperature or some actually whatever condition, so they developed the enzyme such a way is a fit to that process, industrial process. Like that, the simvastatin, montelukast, and sitagliptin, and atorvastatin also they developed, and all this process got the presidential USA presidential award in green chemistry. So later slides we will see those things, and go for the next slide. Next slide, please. Hello.
It's not moving actually. There is a problem here. Sirisha madam. The slide is actually not moving here.
నెట్ స్లో ఉందా?
నెట్ బానే ఉంది మ్యామ్.
Yeah. And furthermore, actually, why it has many advantages. Biocat has many advantages because enzymes are very efficient chiral catalysts, and the beauty of enzyme is like that they can show very highly specific chemoselectivity and regioselective and NH selectivity. If you do chemical reaction, if it is NH is there, OH is there, and perform the acylation in a NH as well as OH, it will both take place. But in enzymes, if it is chemically, if you want to do in NH only it will take place, and OH only take place, like that. And if regio selectivity, we all know in glycolysis pathway, glucose has 6 hydroxy groups, but phosphorylation take place in one position. But so that much regio specific is there. If it is a chemo and enhanced today, single enantiomer only resolute that one. So this is a very beauty of enzyme. This is a very fundamental, very good feature of that enzymes. That's why we can perform very good in reactions by catalyst. And these reactions, however, we can perform the reactions at just room temperature. There's no need, no required of the pressure, all these things. Just we can room temperature only we can do, and atmospheric pressure. There is no need for any pressure, all these things. And broad spectrum of reaction and environmentally acceptable, biodegradable, and these are the renewable resource of the enzymes are renewable resource and less expensive. As I go for the next slide. And you all know very well, because nowadays green chemistry, all heard about green chemistry and the 12 principles. And the biocatalysis, one is the thing is that all the 12 principles is aligned with all the 12 green principles. That's why you just see the in the red this type productively. So there is no need less waste, prevent the waste generation is very less when we use the enzymes, and these are the renewable resource from microbes. So we can regenerate the microbes and we can produce and there is no need for any derivatization or protection, deprotection. Actually, we'll see one molnupiravir process in future slide. I mean, before it will come the slide, next slide, not next, but before there, no protection, no protection, deprotection is required, and is a safe. We perform the reaction in safe and catalytic amount only. We can do the reaction. There is no our reaction can perform at ambient temperature and pressure, and you can monitor, and as well as very few auxiliaries. If it is lipase, no need for the auxiliaries. Hydrolysis, most of the hydrolysis, no need for the auxiliaries. Just water is sufficient for that hydrolysis. And when we do esterification, something is that there is without water. But in case of the CRED and some transaminase, there just we need to give some cofactors like that. And there, when we use the enzymes and E factor also very minimal V factor, and most of the feed converted into the product, yield is very high, and these are the no toxic and is a safer, so safer. So these are the 12 green principles is aligned with the 12 green principles. And so coming to the enzyme classification, you all know 6 enzymes are their classification, but oxidative, transferase, hydrolase, and lase, and isomerase. So we can do all these 5 enzymes. Ligase we did not mention here, but oxidative is a very industrially important enzyme, transferase as well hydrolase and lase. Go for the next slides. And these are the reaction. Suppose if the oxidative enzymes is the EC1 given the name, and then you just see the keto to just we can synthesize the chiral alcohol using is the keto reductase enzymes, and then is a keto acid to get this amino acid, and then is the enol. This in is a reduction to get this chiral molecules, and then some aliphatic is there getting to this hydroxylation. So using P450 getting to this hydroxylation, get this chiral, and then styrene to get the epoxides. So this type of reaction we can perform using oxidative enzymes. And the next slide. For the transferase, actually for the transferase, this keto using transaminase get this amine, chiral amine. Chiral amines are very important molecules for the most of the drugs have the integral part of the chiral amines. And then this keto acid to this amino acid, and then styrene to this alcohol, and then the OH to phosphorylation transfer, and aromatic OH to methylation. So these are the transfers groups from here it take place transferase. And the next, the hydrolase in the first if it is O, it is there, then ester. So ester we hydrolysis from ester hydrolysis using from lipase, protease, and esterase. We can do hydrolysis with that one, and then we got this chiral alcohol. If it is a ester, it's a chiral alcohol. If it is a amide, it's a chiral amide. So amide also we can hydrolyze with using lipase, protease, nitrase. So many hydrolase enzymes are there. Then this ester also getting hydrolysis to get the acid, and then hydrogenase, nitrile hydrogenase or nitrile hydrogenase hydrolysis to give the amino acids, and then the diesters. Then just go for the first slide. Before slide. Hello? Yeah. So 16, slide number 16. So the diester actually in slide number 16 is some diesters asymmetrically synthesis. Actually, we developed industry one slide number 16. Yeah. So from the diester here you are seeing diester getting to the monoester acid. So actually we developed a process for pregabalin. So in the later slides we will see that one. And then here if it is an alcohol or amine. If it is there, alcohol is there, then just esterification to get the ester, and amine is there, then amidation will perform. And go for the next slide. So these are the reaction we can perform with using enzymes. Go for the next slides. Where we will get the enzymes actually. So we just now see what are the importance of chiral molecules and biological activity, and then how the FDA and European agency NSA approved the single enantiomer, and what are the different techniques are there. Among them, biocatalysis is a superior technique for to synthesis of this. But we are where we get the enzymes. So definitely first, we need to screening, isolation, identification of here I'm taking lipase as a producing microenzyme for integration of alcohol. So next slide. So actually, lipase, where we will get the microbes from soils only. How we will select the soils? Go for the next slide. Actually, slide some interpretation.
Actually, there is a power cut here. Once I'll stop sharing and share it again. Okay. Is it visible, sir?
Yeah, I'm seeing, but can you keep in this slide
Yeah,
mode?
yes, sir.
Presentation mode.
Okay.
Yeah, good.
Okay, sir.
The next, yeah. So, how we select the micros for the production of enzymes? Definitely, we will isolate from the soil. Actually, for that soil also, we made minimal salt media. So, when we take the soil samples from now here we are taking the example for lipase. Actually, one we are explaining here. Lipase, how where we will get the soil samples? Go to the next slide. So, soil samples, we will get the microbes from soil samples. How we are selecting? Suppose if our interest is the lipase, what are the doing lipase? Lipase, what are doing triglycerides of? Actually, when it is oil, is there some oils of triglycerides? The triglycerides break down to fatty acids. Lipase do this. So triglyceride breakdowns to fatty acids. So how we will get that where we need to isolate the soil samples? So where the oils spilling more, actually suppose where the oil spilling more, then what will happen? Oil is already enriched. Oil is enriched means there the microorganisms they can oil is availability is more. That's why what microorganisms to do that oil they can consumption that oil. How do they consumption? Definitely they can release the microbes release lipase. That's why they break down to that oils and they can consumption and they get the energy. So that's why we thought in that way, and we isolate the soil sample from Amrit Vanaspati company is a Rajpura, nearby Rajpura, one is the oil company. Then and some you everyone knows a screw type oil mill Chakki. Some when we go for the mungpali or something or sunflower, and we'll go to that mill and they can give the oils. No? So their place and the tractor sets the kala tail and oil drums near generators, and we hostel mess also most of the times cooking with oils, and then we clean that place. So we took that different oil spill places soil samples, and that soil samples we bring in our lab and we dissolve, and then we just supernatant of that supernatant just one micro just supernatant just loop just supernatant loop in the actually previous slide we saw that it's minimal salt media and nutrient agar media. We just spread on that agar plate medium, and in that what we added actually tributylene. We add, because in tributylene what we'll do, tributylene with that minimal salt media and agar media, it will shows the white color, like that, white color. But when we actually when we actually add some microliter of that these things, then microbes will grow on that plate. And if it is that microbe is producing the lipase, what will do, it will consumption the tributylene, and the tributylene, the whatever white color, it will form the clear zones. So based on the clear zones, we came to know these are the hydrolase enzymes. So all the hydrolases break down the tributylene. So based on the trizones, tributylene, so based on that clear zone, and as well as the phenotypical characters of that colony. Phenotypical characters means the some microbes gives color, some microbes give their rod shape or cone shape, some different actually some are like spike heel, and then some are like lubricant, like that. So based on that physical appearance, we select those isolated microbes, and then we will do the secondary screening. The primary screening actually MS tributylene separate only we will take. Then go for the next slide. Then this is the procedure how we are going to do. I already explained. Next slide. So in the primary screening, based on their morphological, this also explained. And then next slide. In the secondary screening, again, what we'll do once we get that primary screening, so hydrolase, most of the hydrolase means it may be protease, it may be stage, it may be lipase, or whatever. Therefrom, again, we need to furthermore, whether it is a specific lipase, for that we have to do the para-neutral phenyl palmitate acid. But para-neutral phenyl palmitate, actually it's dissolved in, actually it give you yellow color solution. Where if it is a lipase is there only, it will give yellow. This is very specific to lipase acid. So we confirmation with here from parameter. Next slide. Next slide. And we confirm the isolated microbes as well as some literature also it will be there now. So we will buy from MTCC culture also. In-house other microorganisms, if it is there in our lab, so those also we can perform. And we make from the secondary screening how many microbes are producing the lipase, like that. First primary tributylene assay, then secondary screening of paraphenyl palmitate assay. The next slide. Then we make the tertiary screening. In the tertiary screening, we perform the reaction with our starting mate, our desired compounds. We just perform the reaction. We synthesize the enzymes and we perform the reaction. Once we perform the reaction, then we isolate, and then we will perform HPLC analysis, whether it is converted or not. We can do also TLC. For a chiral purity purpose, we will do HPLC analysis. Next slide. So, and we made these are the microorganisms for actually some commercially also will take enzymes, whatever lipase available, and then isolated states. So these are like that. We made one tertiary screening, the library of microorganisms. So if it is any time we required any molecule conversion, so we took all these enzymes, then we perform the reaction, and we will see the screening, tertiary screening, how much conversion is there, then further improvement where how to do, like that we can further improve the process. So next slide. And then once we'll get any specific very good enzymes, for that we sequence also. Actually we are isolating enzymes now. So for that what we will do, we will submit MTCC, so they can do 16S RNA, and they give actually they make the phylogenetic trees, and where it which species, which genus come, and if it is a new thing, so they will give some specific number MTCC, some numbers they will provide, and they also do some morphological characters, and they give us this is the identified microorganism, and this is a particular species and genesis, and this is a particular number of that one. So in that way they will provide us. So next slide. So in this way, just summarize the primary screening and enzyme activity, the secondary screening, potentially isolation from the nature. Primary screening we got 26, but from the 26, 16 and another MTCC culture 6. So 22 strain in secondary screening. And when we perform with some specific racemical call, we will find 2 microbes are very good for yield and enhanced emmeric excess. Among them, which one is a better one is identified as a Pseudomonas species. So in that way, we'll do. Then once we identified the specific microorganism, for that, actually how we grow that microorganism, we can grow more as well as produce enzyme more. For that, state plot studies, we'll do studies. So next slide. Next slide. In state plot studies, actually, we grow that microorganism and we incubate some specific condition or RPM, and we took the samples from different time intervals. We see the OD of that microorganism, how growth, as well as we perform the enzyme activity of that specific. And we see actually growth actually the enzyme activity is in a triangular and growth is in a circular. Here enzyme activity versus TAM and OD. So you see the growth is take to place growing slowly and up to 72 hours. Maximum growth take place is 72 hours. But enzyme activity, if you see the initial up to 36, 24 or 24, 30 hours, around 30 hours, there is no enzyme activity. But slowly from 36 onwards, enzyme activity shows and it reaches to the 96 hours is the maximum enzyme activity. So like that, we optimize that media components and the incubator, where is the time and temperature also, which temperature is 30 degrees Celsius temperature, the same experiment we perform and OD and enzyme activity we will see, and then temperature is 30 degrees. And then, so pH is a maximum 6 pH is very good for enzyme activity as well as production of growth of the microorganisms. And then agitation also, it's 200 RPM is the best agitation, because agitation is a very good when it is agitation is a very good thing. Mass transfer, it will take place and microbes took that mass and then consumption and then grow as well as produce the enzymes. The next slide. And then inoculum age also, it affects inoculum age for here inoculum age for growth purpose it is not more effect, but enzyme activity purpose is 18 hours is actually started culture and then production culture. So the started culture inoculum age it should be 18 hours. So then we can do the better in that enzyme production. And from the literature, we also know the carbon source as well as nitrogen source also have effects on the microbial growth and enzyme production. So that's why we perform that different carbon source. Among the glucose is the best one as enzyme activity and growth purpose. That's why here we selected as a glucose, and then nitrogen source. The beef extract is a very good for the enzyme activity as well as enzyme production, growth of the microorganism and enzyme production. So beef extract is good. So we selected like the temperature, all these things. Go for next slide. So in that, just you can summarize optimum time for the growth of that 72 hours and the enzyme activity is 96 hours, temperature 30 degrees and pH. So we once we need we process in the sake plus studies. These sake plus studies data we use and we can do in fermentation. Actually, we can't produce more quantity in sake plus. Sake plus we have to do up to actually up to 1 liter or 2 liter. But even coming to the fermentation, next slide. So further, these are the parameters we can use and we do fermentation. In the fermentation, what will happen in fermentation? In the sake plus batch fermenter, actually sake plus is the batch fermenter. This is also batch fermenter, but there we can't maintain pH, temperature, and we cannot monitor dissolved oxygen, all these things. But in fermentation, we monitor dissolved oxygen or glucose concentration as well as aeration, agitation. So we can see because of that probe, we can pH and dissolved oxygen concentration monitored by pH and DO probe. Actually, we can do more study here, actually further studies. Next slide. And we took our total volume and 3 liter, working volume 2.5. Here we did not actually pH, we determined the pH regulation, but here what we did, we adjust the pH initial, but we did not monitor or control the pH. Actually, sometimes we can monitor also pH, but we did not monitor because we did not monitor and we studied. Go for the next slide. So this is the data actually for the different time intervals, and then is the pH, and then enzyme activity, and then weight cell weight, and residual glucose and dissolved oxygen. So but the dissolved oxygen is a circular, so it's a dissolved oxygen from the very actually when we do it's total consumption. Actually, when microbes add the term is a totally consumption dissolved oxygen, and then pH also if slowly it is increases pHs. And then enzyme activity also, when we see the enzyme activity is a triangular shape, slowly it do after 10 hours, 12 hours, around 12 hours is the maximum activity stored in that fermentation. Actually, when we do actually when we do in the state class studies, there actually 72 hours is growth and 96 hours enzyme activity. But here growth also, if you see the growth weight cell weight, if you grow see also here we get maximum growth at around 30 hours, 20 to 30, in between 20 to 30 hours only we see the maximum growth of the microorganisms, and enzyme activity also we see at 12 hours. Later it's decline. Because why it is decline? The pH, we just see the pH. Initially it's a acidic pH, but later slowly it's going to high, so it's go to basic pH. In the basic pH, their enzyme also degraded, microbe growth also inhibited. So in this way we can study fermentation and we improve that. And here the glucose is more playing the role. That's why in the further study actually we took the different concentration of glucose. Next slide. The further study we just take the different concentration of glucose like 12.5, 15, 20 or 30 gram per liter, and we perform the reaction, and there the growth actually 15 gram per liter is best one, and for enzyme activity also 15 gram per liter is best showing. And you see that the dissolved oxygen there is no use different. If you see the each glucose concentration dissolved oxygen the same pattern, and total reducing sugar also it's the same pattern, it's totally reduced, but in the 15 gram per liter is best for these microbes. So in that way, we will study on fermentation, and we develop the enzymes more, and we perform the reaction with that using that enzymes. On the next slide. So actually, up to now, we just what are the we just see the how the chiral biologically important molecules, and then how the US FDA and the European agency give the single enantiomer. Then what are the different technologies there? Then in biocatalysis, then how the isolation of enzymes and the production of enzymes. Then how to perform the biocatalysis exactly conversion of the molecules. Then we'll see then unitrol. One example. So these are the different approaches are there. If you see the B-neutral, actually the B center is the B-neutral, and the route A, route B, route C. So different routes are there, and here we develop a new route for that. Actually, for retrosynthetically, B-neutral. The C is our route. Actually, B and A are previously reported. For that, actually, what we'll do synthesis of this halohydrate, and once we do this halohydrate, we can resolute with the enzymes. So go for the next slide. So for that synthesis of halohydrin, how we synthesize, just take that cyanohydrin, and with the epichlorohydrin presence of cyanohydrin treated with epichlorohydrin presence of base in acetonitrile, and give this glycerol ether. Once we get this glycerol ether ring opening with acetyl chloride, this halohydrin, and this halohydrin is our racemic alcohol. And that halohydrin, actually whatever we shown that tertiary screening library, the huge library, so all library enzymes we screen here to get the conversion of one enantiomer will be as such, and another enantiomer only converted to the ester. So it's a kinetic resolution. Actually, one enantiomer is a fast reacting, another enantiomer is a slow reacting. So it's a kinetic resolution. But ideal condition is the 50%. Theoretically, it's the 50% this and 50% is like, but little bit is varying the things. So once we do the screening, and then once we get that specific enzyme is giving more selective, So that enzyme will took, again we need to screen for organic solvent. Which organic solvent? Here if you see that here acyl donor, lipase and organic solvent. So first initially we screen the lipase, later we screen for organic solvent, or later for again the acyl donors, later temperature and enzyme concentration, substrate concentration. In that manner we'll optimize all the parameters here. Once we optimize, we scale up this reaction, the lipase kinetic resolution reaction, we scale up, and after the scale up, we perform the scale up reaction. Then we took that, we isolate both the enantiomer, this hydroxy enantiomer and then ester enantiomer, and we convert that enantiomer to further drug. So in this manner we will do. And next slide, we'll see actually we see the results from the actually the kinetic resolution, enantioselective kinetic resolution. Here one enzyme is shows the R selective, another enzyme is shows the S selective. Means in this R selective means it will form R ester, and S selective means S ester it will form, and R alkyl is as such. And so in that way we selected CCl and PCl from the screening, complementary both the selectivity. And next slide for different organic solvent. Actually we perform the reaction in for PCl, Pseudomonas epicel lipase with different organic solvent, and CCl for R selective with different organic solvent. Their tertiary butyl methyl ether is so the best based on their enantiomer selectivity of enantiomeric excess of product and enantiomeric excess of substrate. And here also enantiomeric excess of substrate and product. How we will calculate this one? Actually we perform the reaction, and then we once the complete of the reaction, then we isolate that molecule and we run the HPLC. From the AUC, under the AUC, we calculate the enantiomeric substrate as well as product, and then conversion, and as well as enantiomer selectivity also. And here that we selected for this Pseudomonas epicel lipase, tertiary butyl methyl ether, and toluene for CCl. Go for the next slide. And once we selected that one, then reaction time and different time intervals. And here actually 48 hours, the Pseudomonas epicel is 48 hours, enantiomer excess of product and enantiomer excess of substrate and conversion of everything. Later is not that much changes is there. That's why we selected 48 hours for here. And then this CCl plays around, you say 36 hours is a maximum enantiomer excess of product and substrate, and as well as conversions we got. Very good. So next slide. And these are the acyl donors. So different acyl donors also we will do, because they mainly involve lipase and organic solvent acyl donor. So another parameter acyl donor, but in acyl donor both the case vinyl acetate shows the good selectivity enantiomeric sub product 96 and 94, and then convert selectivity of enantiomer. That's why we selected vinyl acetate as a acyl donor. And go for the next slide. And temperature is also effect on that enzyme and all these things. So that's why we did a different temperature, and we found actually 25 degrees Celsius for Pseudomonas epicel lipase, and then actually per Pseudomonas epicel, sorry, this is CCl. This CCl 30 degrees Celsius and PCl for 25 degrees Celsius. We optimized. The next slide. So once we optimize everything, then we perform. So this type of drug we will synthesize. Actually, when we do the scale up reactions, once we get the scale up, we isolate. Actually, isolation. Then we get this both the enantiomer and treat it with this enantiomer 7A to tertiary butylamine. Get this S21A as well as R21A. So both PCl and actually in actually S21A and R21A. So both the enantiomer of the butyl we synthesized. So in this way we synthesized and we published, and as well as we made patents. So go for next slides. And this is the summary we already discussed. Go for the next slides. Next slide, please. And these are the different molecules. Just only butyl, how we did, and then other lipolysine, metoprolol, guanfacine, NC-present, linezolid, arimiprolol, linezolid, and so many other molecules also we synthesized and lipase catalyzed and we published. And next slide. And these are the some leobunyl also we synthesized, propanol also we did. First time we reported. And here the third one is the synthesis of molnupiravir. What we did in synthesis of molnupiravir, actually we see the molnupiravir structure, the ribose there. In this ribose have the 3 hydroxyl group. That's why what patented method is there, that innovator method is like that. So they are first forming hemiacetal formation of this one, this 2-hydroxy. Later only they butyryl ester they form, and then deproduction of that hemiacetal. So it's the 5 steps. But in our enzyme case, only single step only we identified when we perform the reaction. We also got from enzymes, sometimes some enzymes gives only single product, some other gives monoester and diester and triester also given, but among them which one is given the monoester, that we took and we further optimized. And actually there is actually previously it's a 5-step reaction, but we did reduce the 2-step. There is no protection of protection of both the alcohol as well as deproduction, no requirement like that 3-step. And we published this work. And here reduction of the crate, actually we just reducing this rotatidyl start, and we published this work, and this is a tagoprazole intermediate, and this also we did with the lipases. The next slide. These are the some other biocatalysts we did in our lab. Actually, most of the tetralone to tetralone reduction, alpha tetralone, beta tetralone, and benzosporin, we reduce the chiral corresponding chiral molecule, and those are we used for another further drugs, as well as we have another approach also we did the stereo inversion. Actually, here we are showing here is naphthalene. So in stereo inversion, we took the racemic alcohol R and S. R is as such 50% R is as such will be there, but only S will be oxidized to get the estronapthalone. Again, it is reduced to get R naphthalone. So previously, yes, R will be as such like that, and then again it will convert whatever S to R. So we will get this is a stereo inversion approach. A stereo inversion approach also we did. And next slide. So here, this is the process. US, it's not our process, from the literature. How the enzymes evolved. Actually, the sitagliptin. So this is a pro-sitagliptin molecule. This is a pro-sitagliptin to sitagliptin. Chemical route. How is there? First ammonium acetate, so imine formation, then pressure. H2 means the pressure reaction, and carbon treatment, pre-base crystallization. So even though after 4 steps, the 97% E and 79% yield. So this is the chemical route. But Merck and Codex, both company, they started using transaminase, finding for that this molecule, how they did. Actually, they did very well. Actually, they're 99.95 enhanced numeric excess and 92% yield. So how much yield is increasing? Is a huge yield as well as enhanced numeric excess. How they did? Actually, when initially starting the screening, they did not find any suitable transaminase for this conversion of this pro-sitagliptin to sitagliptin. So that's why what they did, the substrate walking technology. They just break down the substrate. From initially half of the molecule they did, and they find just 1-2% of that conversion. So they recognized their catalytic activity. So binding site into that protein, it's difficult. That's why they increase that catalytic binding site. For that, one side they did 3 mutation, another side, so large size and small side. So 2 cavities there, cavity it will form on small, large. So large side they did 3 mutations, and small side they did 1 mutation. Among them, so all 3 mutation, that molecule, this pro-sitagliptin, fit into that one. Later slowly improved the 11 round of evolution. Each round improving, go to the 11 round. Each round they first initially binding that one, then conversion, slowly conversion percentage increase, enhanced selectivity increase, and then DMSO. It's dissolved in it's not dissolved in buffer, so DMSO is required. So DMSO resistant. So like that, and then substrate 100 gram per liter. So for that also again they develop. So in 11 round, around 30 to 33 lakhs experiment something they had done, and they developed the sitagliptin process. And that this is a very classic process, and this process also won the presidential award. And you just see the how many steps reduced just because of 3-4 steps reduced, and energy selectivity is increased, and yield also increased. A very classic process, and this available in Science Journal. If you go through after finishing our class, if you are interested, you can go through this article. Then next slide. The one more process, the chemostatin. This is also one green chemistry presidential award. Actually, the lovastatin is a from microbial biotransformation. They will get lovastatin, and after the hydrolysis of this lovastatin, monocolin J, at that monocolin J to converting the chemostatin in the chemical process, lactonization, protection, acylation, deprotection, and ring opening. So 5 steps are there. And each step, if you think it's a 10%, obviously if it's a chemical process, minimum is a 10% yield loss will be there. So if you think 10% yield loss, so how much yield loss will be there? So it's a huge. That's why. So one enzymes they developed acyl transferase LOD enzymes. Actually, this also Claytech University and Codex and Merck company. So all three together, and they developed this process very excellently. Actually, the whole acyl, this whole molecule. Actually, if you see the lovastatin to simvastatin, is nothing difference. Only one methyl is only difference. Actually, if you see the structure, only one methyl, the side chain, only one methyl is there. But that methyl total acyl transferase they did actually. This is not a natural reaction, but unnatural reaction. They did it here is unnatural reaction using enzymes. So it's a huge development in enzymes. And go for the next slides. One more, the excellent process, and this is also presidential winning award, US presidential winning award, atorvastatin. Actually, in atorvastatin, initially how they did the route one. If you see the route one side, actually is a diketene to get this chlorodiaketone, and that the diketene is CRED. One CRED is enzymes. Get this one CRED is the reduction to get the chiral alcohol. When they get chiral alcohol, then alcohol dehydrogenase transfer to this nitrile group, and then they can do with actually another CRED. So in this place, 3 enzymes to get this atorvastatin side chain, and the route 2 is nitrylase, again CRED, you get this side chain. But actually, if you see the single molecules in the different enzymes, actually can synthesize different approaches. So they get patent from this route also, the route 2 also. Then later they developed route 3. Here just aldolase enzyme. So chloroaldehyde and aldehyde get this side chain and converting to the atorvastatin. So route 3 is a very efficient process, and that aldolase enzymes they develop. This is also a natural reaction. Actually, aldolase is present, but not done this type of molecules, but they did in this way. Actually, getting the that's why this also got the presidential award, Merck as well as Codex company. The next slide. So in that similar way, in the one pregabalin also, pregabalin is this is cyanodiester. The cyanodiester hydrolyzes getting the monoester, and this monoester further chemical reaction converting the pregabalin. So with hydrolysis is there. Next slide for pregabalin, and then enolate reductase. With enolate reductase is there for pregabalin, just in the cyano monoester here the in is there in reduction take place and getting this one, and later chemical step to get this pregabalin. The next slide, hydrolyze, enolate reductase, and transaminase also. So they reported with transaminase, just omega transaminase getting to this the single step getting the pregabalin. And then next slide, the nitrilase also, the nitrilase. And one more, actually we developed in industrially lipase. So later we will discuss in that process. And next slide. So as of now, we are discussing about actually single enzyme develop the single molecule like that. So here, just recently only published, this is a one molecule recently only published isotravir cascade reaction. How is the cascade reaction? They actually whatever is the starting material to getting the MK8591 is a chemically it's a 16 steps. And each steps, if you think each step chemical, each step reducing the 10% lose the yield. So after 16 steps, how much yield you will get? So very less yield, and that's why just 9 enzymes and the 3-step process. It using enzymes only 3 step only they developed. Using 9 enzymes. Among them, 5 enzymes are only 5 enzymes are remaining 4 enzymes auxiliary enzymes. In the 3 steps they develop, and yield also is a 50, around 51%. The next slides. Next slide. We see here 2-ethynyl glycerol. Here galactose oxidase variant. This is enzymes they evolved, and either catalase and horse peroxidase. Catalase enzymes is like that. It is a regeneration cofactor, and the horse peroxidase keep the copper is a that state. So it will can convert the reaction totally into this one. And once converting this one, here is a pantothenate kinase variant. So the kinase variant is a directed evolution, or the estate kinase is a auxiliary enzyme for cofactor regeneration, phosphorylate cofactor regeneration. So this is the first step and the second step, and the remaining 3 steps are in equilibrium. Is only one step, but it is equilibrium. Like deoxyribose 5-phosphate aldose variant and phosphatase pentose mutase variant and purine. So here just cyclize this form, and then phosphorylation change from 5 position phosphorylation to the 1 position, and there the nucleotic this nucleoside purine nucleoside addition to this phosphorylation get this etc. So this is 3 steps, 3 enzymatic steps, and here only 5 enzymes are evolved. Remaining enzymes are already reported things they can use, and they synthesize this one 51% old. So this much capable how the biocatalysis, and this is the very beautiful process. And not only this, so many are there, but I just took one example here. And next slide. So the telescope way given the concurrent step and telescope step. And the next slide. Next slide. This is a pre-gobbling process. Actually, we developed this process in industry. Here, actually, pre-gobbling, how we see that hydrolysis enzymes are there, cyanodiester, and then nitrate enzymes are there, indictor is there, transaminase is there, and then one more process also we developed using CalB. So cyanodiester, so asymmetric synthesis way, PG and DME to get SPG and MME, and then later the ammonia reaction, get this amidation, then Hoffman degradation, get this a pregabalin. So this is the industrial process. Vinita, do we have time to discuss more? Hello?
Okay, sir, please continue, sir.
5 minutes more you have, sir.
Oh, 5 minutes. Okay. So actually, industry. So when I joined in industry, actually how it is. So they recruited us, and based on our performance, as well as once we recruited, they will provide us training. So when we recruited in that, they will train in this way. Actually, each department they will send in HR department, then safety division, then QA division, QC division. So then we will go there and we will see the SOP, all their SOP we will read, and they will keep exam also. So for good process, they will train in this way, training and professional development. They will keep, if it is any modification is there, regularly they will keep some conference in that way. They will took the classes, and they will say, so these are the changes or modifications took place. So like that. And the next slide. When we buy the equipment, what we'll do actually, when we buy the equipment, actually we buy, then we design qualification and installation. And once we do the installation, the performance qualification, operation qualification, and all can we do, then we will maintain their calibration, calibration time and everything, and keep the good records for the equipment. So our results will not deviate. So next slide. So these are the, when we perform that validation, all these things, it should be reliable, accuracy, precision, and calibration, linearity. It will all coming towards. And then if it is a compile all these things, then we can go for further process. Actually, when we do the lab scale reaction in the lab, also perform the 3-time batches, see the yield, consistency yield is there, concentration reaction, duration of the same duration it will form. And then suppose if it is impurity will form in each step, how the impurity profile is there. Each time, each batches impurity profile is similar. So we will see in batch process API Kilo Lab, as well as in ADL, they develop their methods, and there also they will say check that reliability accuracy of that method. So continuously inject and everything, all know validation that methods. And once the validation, the precision accuracy in the range only, they can fix that one, and then we will perform the reactions. And further, so lab scale to pilot scale to kilo to pilot to plant scale. So next slide. So actually in industry, how it will be done when we are doing the reactions. Suppose it will take the case study of pregabalin. So pregabalin, we are getting the DME. DME will actually, suppose only one vendor will depend. Sometimes those vendor will not provide in future. So what will happen? Our process will stop? No, nothing like that. So that's why we'll take different vendors. So where are the different sources of that raw materials are there? We will find out those raw materials, and then we do vendor qualification. Actually, where how their impurity profile is there, their impurity profile effect on that our process. Is it in the process impurities or in carry forward impurities, and how detection also every time is how much is it is there, and final how it is elevated effect. So like that, so vendor qualification, then we will see for one molecule. So one intermediate we can see if you want to buy. So take two three different sources, and we will do vendor qualification, lab as well as kilo and plant and everything. We'll keep the records. And if next slide, may maintain the management and the quality control purpose. QC people will be there and QA people will be there. They will take care about that from ADL to QC, transfer that methods, and from TT people is there, technology transfer people, from our lab to kilo lab to pilot lab to plant. So they will take care about all the safety people will be there, and management, all the peoples will there, and then each validation, method verification, and they can further go further plant validation, all these things are commercialization. Go for next slide. And if it is any mistake will happen, any problem is there, so what is where it is a root causes of that mistake, and then corrective action, the preventive action that took place, and they will make the SOP, and then train them, and they rectify all things. The next slide. Yes. So actually audit also will be there, internal audit as well as external audit there in industry. So documentation they keep very nicely, as well as if it is anything mistake also happen, then they will raise incident, and then that incident again they need to do correct action, preventive action, and all the documents, the train, everything they keep very properly. So next slide. If it is anything there, again for that, so again they will do, if it is a problem is there, again rectify that solution, then preventive action, corrective action, and then make the perfect to go towards the process. So next. Okay, next slide. Yeah, whenever we are doing reaction, always they will tell us to safety is important, and the safety people will come and check whether we are following the safety. And whenever you are also doing some reaction, all these things, the safety is very important. In our IACD also, safety is the first priority, as well as in industries also, safety is a priority. So next slides. Yeah, next slide. Yeah, thank you. If you have any, okay, done. So any doubt or questions?
Yeah, thank you, sir. Dear delegates, the session is now open for the questionnaire. Any questions? Actually, sir, you have given a brief, vast information about the importance of chirality and green synthesis and effects of certain parameters like pH, temperature, agitation, growth on the enzyme activity. And in relation to the industry also, how can the quality assurance people work over there, sir?
Quality assurance people will actually they will do the internal audit actually. How they will do now? They will come to ADL. Everything will be recorded. If you carry the experiment is recorded or not. They will check and go to the ADL also. If they are doing any analysis, there each experiment. So actually, in when I was working in CIPLA, there is a electronic notebooks. So if you do anything, we have to record it in electronic notebook. Like that, ADL people also electronic notebook. Once we raise the one experiment, so each things we have to fill and we need to go conclusion also in the certain that given time period only. We complete that everything. Actually, conclusion also we need to finish experiment also. So today we will start experiments within week. You have to finish your experiment and in your review, whoever you are reviewing that experiment, that one also if it is a review and if it is anything is mistake is there, so review will say something revision, so we need to do that revision again, we need to submit and it's closed. So those things whether R&D people are doing and ADL people are doing, and once we develop the process and from R&D and ADL, definitely it will go for R&D to kilo lab or pilot lab or plant, but ADL to QC. So each documentation transferred very well like that. They will check those documentation if it is everything is okay, and the assurance to how the regulatory says the impurity profile of that molecules, if it is in the limit regulatory as per according to regulatory limit only they fixed specification, or if it is specification fixed in that way in that direction, if it is a 0.1% or more impurity is there, they characterize that impurity, or it is any anything is effect is there. So like that the QC peoples play a very good role in internal audit way, and they will guide us if it is anything is going beyond the regulatory or according to norms, it will not follow the WHO guidelines, all these things. If it is okay our process, then they will say okay. Otherwise, they will comment on that. Again, we need to rectify it, and they will check in that manner. They will check. It's a very good, very good work.
Thank you, sir. There is one question from the audience.
Yeah, one more. Actually, QC people says they also take. Actually, if it is something is happen, some problem will be there. So if you incident raise, so problem is there. So they will take care about the actually the QC people. Most of the times QC will raise the incident. Actually, QC sometimes make the problems like something mistake is there. So they will raise the incident. So the incident raise the properly they raise, and that properly they find out that cause of the root cause of that problem, and then they train whoever did that mistakes, they train for them, and the train documentations are there, are that COPA and preventive action they took. So in that manner also they will take care about not only QC and safety also they will check. So it's a very good job. Tell me.
Thank you, sir. There is a question asked by Dr. S. Hemalata, ma'am. Can lipase catalyze the selectively for one isomer leaving the other isomer?
Yeah, sure. Why not? Actually, we saw so many examples here. No? Actually, butynolol, ubinolol, propranolol, and even we saw the molnupiravir. So many examples are there. Definitely, it will do. And not only we did, but so many literature also is there.
Yes, sir. Thank you. Any more questions? Okay, sir. Thank you for taking out your time and giving us such an enlightening lecture on the topic towards sustainability, designing and effective cascade synthesis by using different enzymes. We got to know about different ways how which the chiral molecules will be acting, and what is the importance of different enantiomers, what is the effect caused by different types of enantiomers, and how the green synthesis, how the synthesis utilizing the biocatalyst will be useful over the normal chemical synthesis That means the side effects or whatever the hazardous chemicals which we use during the regular synthesis can be avoided using this biocatalyst. Thank you for giving your valuable information to our staff and students here over, sir. Thank you. Thank you once again.
Okay. If you have any doubts, you can contact, no problem.
Oh, okay, sure, sir. Okay, thank you, sir.
So shall I close the session, Vinita?
There is one more question, Vinita madam.
Yes,
There is
ma'am.
one more question. You can see in the chat bar.
Okay. So, okay, sir. There is one more question. For selective action, what conditions need to be maintained in chiral selectivity?
I'm not got. What is question?
For selective action of an enzyme, what conditions need to be maintained in chiral selectivity?
Selective, actually conditions, it depends on enzyme by enzyme. Suppose if you take hydrolase, lipase, so there actually mainly play the role enzyme as well as organic solvent and acyl donor. In case of crate, there it will play buffers and enzymes, and in case of transaminase, there will play a cofactor PLP, buffer, and amide donors. So it's a depends on enzymes and then molecules and case to case. It's varying. So it is not a specific, but it is varying, and we just do trial and error method only, and we just do trial and error, some scientifically and rationally, and some trial and error to fix the selectivity of these things.
Thank you, sir.
Any
Okay.
more questions?
No more, ma'am.
No more. Okay. Shall I?
Okay, we are ending the session, sir. Thank you again, once again.
Okay, thank you, Anita. Thank you, your principal, ma'am, actually giving opportunity.
Yeah, thank you, sir. Actually, in the
Yeah.
morning itself, 11:30, I want to speak to you once. Thank you once again, and thanks to Vinita madam also.
Thank you, ma'am.
It's totally pure chemistry. I mean, after a long gap, I am going through the chemistry slides actually. I am of pharmaceutics background as well as quality assurance. Thank you once again, and we'll call immediately, means as soon as possible, to our college, sir, to visit as well as to interact with our M-Pharm students.
Sure, madam, sure, no problem. As your wish. Okay, thank you, ma'am. Thank you so much.
Thank you, sir.
Thank you, sir.
Okay. So,
Okay,
so
ma'am.
dear delegates, with this, we came to the end of day 4 of our online FDP program. We would like to extend our sincere gratitude to our distinguished speakers, Mr. B. Narendra Kumar and Dr. Dinga Banot, for sharing their valuable insights and expertise with us. Thank you. Dear delegates, we'll meet again tomorrow morning at 10 a.m. Tomorrow morning session starts at 10 a.m. Please note that. Okay. With this, we'll end the session. Thank you, one and all.
Okay, ma'am. Bye.